Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is with Iain Drummond, who is using zebrafish as a model to figure out organ development and regeneration. Iain is a professor and director of the Davis Center for Aging and Regeneration at the Mount Desert Island Biological Laboratory in Bar Harbor, Maine. I learned a lot during our conversation about kidneys and regeneration, but I also learned a little history. MDI Biolabs is the oldest research institution on Mount Desert. I really enjoyed hearing about Iain's work, including his leadership and collaboration with the Rebuilding a Kidney Consortium. I've included a link to that work in the show notes. Iain, welcome to the Maine Science Podcast.
Well, thanks for having me.
I know you work with zebrafish and regeneration. I was thinking you could give us a super quick overview of that part, and then I'm going to make you do a little bit of this is your life. Back in the day, how'd you get here kind of thing?
All right, sure. Zebrafish are really useful because, one, they're optically transparent. You can watch cells march around and do different things during development. But more and more lately, people are keying into the fact that fish and other cold-blooded vertebrates really have remarkable powers of regeneration. So if you injure a kidney in an adult fish, they can grow new kidney structures. And this, unfortunately, is something we can't do, and we'd like to be able to do that. And so the real game for us is to figure out how fish do it, how similar is it to what we do in embryos to make our kidneys initially, and then can we take advantage of what the fish does or our knowledge about that and apply it to humans? Can we engineer new tissue generation in humans? And that's the basic driving force for just about everything we do.
I remember touring a zebrafish lab a couple of years ago, and I thought the number that they said was that zebrafish and humans share something like 97% of our same genes, so that it is more translatable to look at zebrafish as a model than most people would expect. Is that true?
Yeah, and that actually extends further down the evolutionary tree, and even things yeast and C. elegans have similar genes to humans. It's basically the rationale for using any model is that you can do lots of interesting experiments in very controlled ways and learn things you can't learn from studying humans. Humans are kind of a retrospective look. You can't modify genomes and see what happens. So with experimental models like the fish, you can do all sorts of genome modifications, mutate genes or add new genes, and test what they do. And that really informs us a lot about human biology, because then we can look for similar genes in human disease. My lab has targeted genes in zebrafish and later found out that there are mutations in human patient populations that cause the same defects in humans that we see in fish. And by studying them in fish, we can identify the genes that have mutations in humans.
So a little bit back in time, I know, like me, you're not a native Mainer. And I also saw a little bit that you went to Union College, which made me smile. I grew up in Colony, so just down the road from there.
Down the road.
And then Berkeley, so the other coast. I saw that, and then yeah. I think you've settled on the East Coast. So maybe if you could give us how did you get into biology, and then a deeper dive into the genetics. And you've seen a pretty vast change in the world of genetics and how we study it in your career. So maybe you could give us a little bit of that as well.
Yeah, I'd say my first deep dive into genetics or understanding the significance of genetics was imparted by a high school biology teacher, Mrs. Platty, Sue Platty, who ran the AP Bio at Wellesley High School, where I went to high school. And she really left us alone to formulate hypotheses about observations. And it was the first time in my education that I saw, that my ideas might matter. Not sure they really did, but she at least instilled that idea that our ideas should matter, and let us explore the phenomena put in front of us. And she had a model of bacterial interaction and mutant bacteria, and really understanding how genes impact enzymatic activity and enzymatic pathways. But of course, I knew nothing about that starting in her class, and she led us to literature and let us explore on our own and ultimately write our own papers on it. And so that, after a youth of, playing with anthills and studying plants and, having a chemistry set, that sort of consolidated my ambition and confidence really in being a scientist. So I felt that launched me towards college. So I went to Union College, as you mentioned, and there I got involved in research as well. I think once you're bitten with the research bug, you really just want to keep doing it. And so fortunately, there was research projects ongoing in labs at Union College. I studied the ecological impact of dredging a lake on fish disease and physiology. So they were cleaning out some lakes in upstate New York, and we compared the fish in those lakes to ones that were in pristine Adirondack lakes. And we found stuff, their liver was full of age pigment and signs of stress. So there, again, it was, exploration and looking at your own data and trying to believe it, or having the faith to believe it and make conclusions from it. And so that was another sort of foundational experience for me. And then I took some time off after that. I didn't want to I was convinced I wanted to go to grad school, but I really didn't know what I wanted to do. So I worked for 3 years as a technician. And these days, actually, most people who get accepted to good graduate programs go out and have some work experience in a lab. It's kind of a rite of passage, I suppose. Basically, it helps show commitment to the field, and you understand exactly what working in a lab is. So it really helps you get into grad school, bottom line. And I got accepted at Berkeley because by that point, I had really developed an interest in how cells communicate and how principles of neuroscience might apply to non-neural cells. So principles of neuroscience are electrical activity, ion conductances, ion signaling in cells. And that was sort of a brand new idea that non-neuronal cells could use the same kind of mechanisms to regulate their behavior or signaling. And so that was a blessing that there was a professor, Rick Steinhardt at Berkeley, who took me in and educated me. And so that's how I got to grad school. And it was at a time when genetics was just blooming and recombinant DNA technology was just blooming. And honestly, I had education background in it, but I had no lab experience in it. And so another sort of foundational passage for me was going to Woods Hole, to the marine biology lab, and interacting with people, doing molecular biology, doing protein analysis, doing all the running of gels and things that I sort of craved but never had experience in doing. And so that short 2.5 month stay sort of changed the shape of my PhD thesis, so that when I came back to Berkeley, I could apply everything I learned and have new tools to explore the phenomena of cell communication that I wanted to do. So I think those things are real important, education, hands-on experience, driving your education with your own interest and ambition, and then it really sticks with you and you use it. And so I think those are the kind of experiences we try to foster where I am now. We are very much a hands-on institution who encourages young scientists to come join us with little or no background in what we do, but with a willingness to get their hands on a pipetter or mate some zebrafish, look at them in a microscope, because that's where you really gain traction in research and start trusting your eyes and, start thinking for yourself. And I think that is what we specialize in at MDIBL.
So you said you did cell communication for your PhD, and I know now you're doing just really cool regeneration work, and maybe I'll just leave it at that. If you could explain what that is in better terms than I could possibly do, that would be really helpful. And then fold that into what you're working on specifically with kidneys?
Yeah, well, cells communicate all the time. So during formation of an embryo, cells have to talk to each other to understand how to pattern into different cell types and different tissues. And for instance, you could take a sea urchin embryo and draw a string around it and make two of them. You don't get half sea urchins. You get two complete sea urchins. And that's because the cells during their development talk to each other and regulate who they are and where they should be. And that whole process is just part of all of life, actually, of how cells and organisms function. They talk to each other. So in development, it's particularly interesting in regeneration because you have to proportion different cells and get them to move to different places and create a functional limb or a functional kidney. So what we're trying to do with the fish is kind of replicate that or understand how that process is replicated in an adult regenerating kidney. So during an embryo formation, during kidney formation in humans, and during fish development, cells talk to each other. They send signals. The signals might cause cells to migrate to a certain place. They might cause cells to start sticking to each other. They might cause cells to change their shape and polarize to take a form that's part of a mature organ. And so figuring out what the signals are, what stimulates them, why are they in certain places to recruit cells to certain places, how cells respond to those signals by growing into mature tissues. That is just the broadest question in developmental biology and now in regenerative medicine. So in a nutshell, what we've discovered is that there are what are called progenitor cells or stem cells that exist in a fish kidney, that when the fish is injured, there are signals that stimulate them to proliferate, migrate, aggregate, form new little structures that grow out like an expanding tube, and then plumb into the existing tubular architecture of a kidney so that you get a new functional unit of a kidney. It's called a nephron. It's basically a blood filter. But to have that work, you have to have a tube and you have to have a filter at the one end and an output at the other end. And so all those processes are controlled by cell signaling and cell communication. And what we're trying to do, the outcome of our work in fish would be that if we can figure out how to fish adds a new nephron to an existing kidney, we could take advantage of all the stem cell biology and organoid development and human biology to try and figure out how to engraft those into human kidney patients. And that's my involvement in this larger consortium called Rebuilding a Kidney at the NIDDK, the National Institutes of Health. I'm a firm believer that these basic model organisms can teach us something that we can apply to human health, and that these basic model organisms really aren't that different from us in terms of how we initially formed our kidney. Fish preserve those mechanisms into the adult. And if we can just figure out how to turn them on in a human or engineer them in a human, it's not a big leap to imagine getting engraftment of new tissue in human organs.
All right, so I have two questions. The description that you have for the fish and the kidneys, that is similar to what people think about with a starfish growing a new limb, right?
In essence.
I mean, I realize I'm very broad here, not...
So I don't know if it's a stem cell mediated response in the starfish, so I don't want to just jump in because I'm not an expert in that. The concept of, I can talk more about limb regeneration in salamanders.
Okay, great. Let's do that one. So it's the same idea as that?
So in both, you have the concept of cells that have what are called multipotency, basically cells that are resting or sitting there, and at injury, they are stimulated to become many different cell types. And so in limb regeneration, fibroblasts, the Prayag Murawala at the MDIBL is working on how fibroblasts can become bone, cartilage, tendon, when limbs regenerate. So that's what's called multipotency. And that's a very powerful concept that, you can have one cell come to many things. And so the goal in both my work and in Prague's work and other people at MDIBL would be to understand what signals stimulate that, how you decide how many bone cells to make, or in my case, how many new nephrons to make, and what are the actual, communicators of that response. So basically, somebody has to make a signal to say, get on with it, start making more cells, be this cell or that cell. And that's a very potent place to start, because those are key, sort of high in the hierarchy kind of information for the whole process of regeneration. What kicks it off? Because often once things are started, there's all this self-regulation or communication between cells that will end up forming organ tissue. But if you can figure out what initiates it and how to kick the ball on, then those are very applicable, I think, to regenerative medicine approaches.
My second question is, do zebrafish only do this with kidneys, or are there other organs as well?
No, there's many organs. MDIBL, Iain has worked at MDIBL to show the heart regenerates in zebrafish. Others show liver. Liver's a pretty good regenerator in humans as well, but it's very active in zebrafish. The fin is one that's very accessible. The fin in fish will regenerate if you just snip it off. And it's quite remarkable. It's analogous to the limb regeneration in salamanders. So it's a great model because it's right in the outside of the fish, and you can monitor it visually and understand the process. So that's another tissue, but many tissues in the fish will regenerate. And I'd say there's two main kind of ideas in regenerative medicine or organ regeneration. One would be that there are cells that are set aside in an adult organ that are capable of forming many types of cells, or forming new muscle, for instance. So satellite cells are cells that sit adjacent to existing muscles, and that when the muscle is damaged, they start proliferating and form new myotubes and incorporate into muscle and restore muscle function. And that's an ongoing thing throughout life. Even when we exercise, we're inducing new muscle formation. So that's the concept of a set-aside cell. You keep it in reserve, and the issues with that are if you run out of those cells or you deplete them, then you lose your regenerative capacity. And that's sort of an issue in aging, is how does aging impact our regenerative abilities? And then a second major concept in regenerative medicine is that cells can de-differentiate. What that means is you might have a cell that's functioning as a connective tissue cell. And then once you injure it or cause a wound, that cell is stimulated to proliferate and then form new tissue and form different cell types. But the concept is different there, because it's saying that an existing cell that's functioning in a tissue is not set aside in a reserve someplace. It's actually part of the tissue that's working, can proliferate and restore the overall function of the organ. So the liver is a good example of this. You can cut off a lobe of the liver, and it's the actual liver cells that start proliferating and growing into and replace that missing shape of the liver. But both concepts involve this idea of multipotency. The cell can become many cell types. They incorporate the concept of cell communication. How do I know a lobe was cut off? How do I know a muscle was damaged? There are signals that go to the cells and tell them to get activated. So those are some key concepts.
And are you finding with the different, I realize that you're looking mostly at kidneys, and we can go to that in a second, but how much overlap is there between how each of these cells do this organ-wise? Like, is it different? Is it a different process for the heart? You said the liver is different from muscle cells, for example, because it doesn't have something in reserve. It's the same cell. Like, how much translation is there? If you learn, say, for example, you learn about the kidney, can you then say, well, I totally know how this is happening in the spleen?
Well, I mean, to be fair, you'd have to say that all the contexts are really different, but what's shared are key signaling pathways. So there's kind of it's not a joke, but it's kind of an oversimplification that there's basically 5 major signals. You have FGFs, Wnt, sonic hedgehog, bone morphogenetic proteins, and I'm forgetting something important. But so what you see are the reuse over and over of ligands, secreted like growth factor hormone-like substances, and receptors that would understand that or translate that signal into a response in a cell. And so I think through the process of evolution, we've just reused these signaling ligand receptor systems. But once you put them in other contexts, they have other meanings or they have other outputs, right? So in a sense, yes, we can say, I know how the gut regenerates. It's an epithelium. Let's look for those molecules in a kidney. And that's actually been very useful. But once you get down into which of the large family of molecules they are, and more importantly, what is the output of their signaling, that's where you get into organ-specific type responses, and that you really have to understand. But you can learn a lot from looking at signaling systems and in sort of a metaphorical way between organs.
As an aside, the idea of something called sonic hedgehog as a critical feature is pretty awesome. So
it was named based on a Drosophila mutant called hedgehog that has these sort of runted larvae that look, when they're spiky and they have extra cuticle little protrusions and they look like hedgehogs. And then when it came to finding, so there's an example where a basic model system, the Drosophila, educated everyone working on vertebrates, humans, that the same molecules could play out or have amazing developmental impacts on both a fruit fly larva and a human. So it's the same, it was cloned based on homology to Drosophila, but there were at least 3 of them in humans. And so they just gave them fanciful names. There was desert hedgehog, sonic hedgehog, and Indian hedgehog. And I think so that's I guess one of the fun parts in biology. You can name things still and
well, it certainly gets your attention, right? I mean, it stuck out for me.
Part of it. Yeah. I was informed that when I was cloning all these zebrafish cystic mutants in the kidney, that I had to have a theme. You've got 18 mutants. We want to see a theme in your name. So they were all based on bubblegum. So we have Double Bubble is a mutant we've worked on. Fleer is another one we've cloned. And I guess it's a trademark violation. But anyway, we do that.
So why kidneys for you? Is it purely advantageous based on your interest? Is it the fact that, and I know that this part's true for, kidney disease is pretty brutal, not just in the US, but worldwide. There's real issues with I hear stories and anecdotal information about the hours that people spend doing dialysis and things like that. So some type of way to rectify that. And I do realize that you're doing basic research to figure it out. So I don't expect that we're going to go from what you say to next year, a magic solution for everyone. But I am curious why you pick kidneys and what you see as kind of the long-term goal of what you're doing.
Well, I have to be honest with you. I didn't go through college thinking I'd work on kidneys. I think what happens as you mature as a scientist is you look for where there are good opportunities to solve complex problems. And in the early 90s, when I was a postdoc, there really weren't a lot of people doing kidney. And it is one of the most complex organs. The kidney function requires, at least 30 different cell types. It's a very intricate process. There's a lot of physiology obviously involved. So all those things, I had a background in physiology, and I also had a background in cell development and biology and cell biology. And so there was an opportunity there for sort of an underpopulated field. So those are fun times because just about anything you discover is, useful and interesting. And also at that time, the zebrafish was just being explored as a way to study organ function through genetics. And I sort of latched onto that because I was in love with these transparent organisms where I could, really see cells performing and behaving in live embryos, and that no one else was working on the kidney in fish. And so I defined sort of the basic geography of kidney development in the fish, and also the impact of different gene mutations that were both similar to humans and also went into unexplored territory and then circled back to humans. And so that's really where the power of the models is to really go beyond what's known, use them for basic platforms of discovery, and then translate them back. And we've been very successful in doing that. And so that consolidated my commitment to the kidney. And also it brought on a field of people who would use it for application to, human disease. And so over the last 15 years or so, we've investigated polycystic kidney disease, focal segmental glomerular sclerosis, and ciliopathies. These are basic problems of human disease. And the fish has those features and also what we call phenotypes, which are the outcomes of gene mutations that replicate disease. And so all those things really made me think it was worth working on. And so most recently, well, actually not recently for me, for the last 12 years or so, we've been working on regeneration. And it's a bit of an uphill argument when you work on something in a fish that humans don't do. So in terms of funding, we were very lucky to have foundation support from the Harvard Stem Cell Institute, for instance, to get these studies off the ground looking at regeneration. And now I think people appreciate that there are links between, fish kidney regeneration and the potential for human kidney regeneration or engraftment. So you just have to push and you have to keep working, and you really are indebted to your colleagues and also the tools and approaches that we can use at MDIBL to study these things. We've had a major investment in microscopy, which really helps us. We have a really good core bioinformatics group that helps us interpret the results we're getting. So in terms of how I got into the kidney, it's sort of a long-winded answer, but I think there was a great opportunity and a great potential to use the fish as a platform to really push the field forward.
It's another good example of the value of basic research, which I think we're seeing play out in front of us with the COVID vaccine results. The mRNA vaccine didn't just come out of the blue. That has been a foundational piece of basic research for quite some time now. So what you're saying about, getting funding from a private source to get this jump started seems like it was pretty critical.
It is. And I think there's so much evidence that basic research changes the world. I just want to keep sending that message. If you look at what has really fundamentally changed our approaches, it was work that was done on innate immunity of bacteria. Okay, so Doudna discovered and Carpenters discovered CRISPR-Cas. And that was, who really cares about how bacteria fight off, phage viruses? That's not my life. But that understanding in basic molecular biology is allowing people to be cured of sickle cell disease. So that's pretty amazing.
It didn't take that long, right? That's the thing. We're talking about easily within our lifetimes where that's been true.
Yeah. And so that just a plug for MDIBL is that we are committed to basic model organisms and basic research. And we're certainly happy and motivated to translate it to human disease. But we think that the engine for advancing science is still in basic experimental organisms, where we can do really, really well-controlled experiments and genetic manipulations that let us really probe the deepest aspects of cell biology and regenerative medicine and aging as well.
Emily Burke sent me some information on you, and then I did some more searching because I wanted to make sure I didn't miss anything. And the Rebuilding a Kidney Consortium that you're part of seems to me that it's both a mix of basic research and starting to get into much more applied's not quite the right word because it's not there yet. But one of the names that jumped out at me was Leif Oxberg, who spoke. I think it's Oxberg.
Yeah.
Yes, he spoke at the Science Festival in 2016 when he was still at Maine Medical, and gave this amazing talk about his work with kidney regeneration, which is as cool as yours and totally different.
Yeah, he and I are on the same team at Rebuild a Kidney. So we applied together for a grant proposal and got it. And what we're doing together is we're taking advantage of everything we've learned in the fish and trying to engineer it in human and mice to start with. And so Leif is a key driver of what's called organoid technology, taking stem cells and converting them into kidney tissue. And then we're understanding now that the same signals that do that are driving kidney regeneration in zebrafish. And it's illuminating for us that there's just a few key steps we have to bridge, we feel, to get engraftment of organoid tissue into an existing adult kidney. And so the Rebuild a Kidney Consortium is committed to that goal of dealing with this problem of organ replacement or renal replacement, because it is a big problem. The sad fact is that there's not enough organs to go around for transplant. Most people who hit end-stage renal disease are destined for dialysis for the rest of their lives. So it's not great. So we have to do something to improve that. So that's what the Rebuild a Kidney Consortium was founded to address: is how do you make new kidneys? How do you make new devices? And that is a broad consortium that incorporates me working in fish to Leif working in human kidneys, human stem cell derived kidney tissue, bioengineers who are 3D printing matrices to incorporate tissue into, trying to connect tissues to have functional inputs and outputs. And beyond that, a major push into how can we promote the endogenous cells or the injured kidney? How can we promote repair of itself before you even get to end-stage renal disease? And so understanding pathways that cells take during repair, whether they're productive repair or non-productive, sort of senescent is what it's called, where cells are sort of non-dividing and sitting there actually being kind of nasty. So how do you promote productive repair? So that there's many aspects to regenerative medicine of the kidney, and this Rebuild a Kidney Consortium is committed to this array of choices. And I'm asked when's my kidney coming, But I think I'd be very surprised if there weren't a major breakthrough in the next 10 years in terms of engrafting tissue, promoting productive repair. And that's because just the rate of science, how it's going these days, how many tools we have to manipulate, how many tools we have to look at cells at a single cell resolution and understand how their RNAs are different, even between cells that look alike, how they're really molecularly different and how they communicate to each other. So there's a great potential these days with all the tools, and it's just a commitment of time, energy, and people, I think, and we'll do it.
This leads to the idea that you're going to make these advances because of all these different areas that you're all looking at as well, right? No one's in a vacuum here.
It's really more than one lab can hope to achieve. And because we need expertise in all elements. And so it's great for me to continue doing what I'm good at, which is zebrafish genetics and discovering how fish do it. And it's great having a colleague like Leif and also another colleague, Denise Marciano at UT Southwestern in Texas, who's doing all the same sorts of experiments in cell culture models of epithelia cells. And so you can discover something in one system, but what you really want to see is how does it apply broadly, and how can we take advantage of mice and cell culture to do very specific sorts of experiments that the other systems aren't good for. And so you build kind of a body of work based on multiple different approaches to the same problem. And when you have multiple successes using this approach, you really do trust the data that it's relevant and broadly interesting and useful.
So one last question, I think one last question, although don't hold me to it. I know you're relatively new to full-time at MDIBL, so kind of welcome, although it seems like you've been coming to MDIBL for quite some time and doing some really great work. You're the director of the Davis Center for Aging and Regeneration, which is, looks like your official title. Can you talk a little bit about what that means and what it is exactly that overall what you're looking to focus on at MDIBL? And I don't mean you personally, but I mean the whole
the vision for the institute.
Yeah.
So I sort of touched on it before. What we specialized in, and historically for the past 100 years, we're the oldest research institution on the island, and what we focused on is the power of comparative analysis for understanding basic biology. So that means looking at different organisms and comparing how they function to get sort of what are the common truths of biology. And for many years, we were comparative physiology, taking advantage of sea creatures, different fish and simple organisms from the ocean. And that's how marine stations actually got their start, was that they had models, in the water outside the lab. But now these days, of course, people bring the models into the lab. We have aquaculture and we grow C. elegans and we have fruit flies. And so the idea of MDIBL remains the same, really, as it's been historically, is to just take advantage of comparative analysis to tackle essential problems in biology and health. So we're focusing on regeneration, we're focusing on aging, we're focusing on concepts of what are pluripotency, how do cells maintain that undifferentiated state. And so as we grow, which is our mission, we recruit new talent to sort of expand our horizon of models to look at for aging and regeneration. We have new initiatives in fish that has a short lifespan, African turquoise killifish. And so we have the facility to build a colony of these fish and take advantage of the fact that they only live for 4 months, which makes it practical to study lifespan extension or health and regeneration in old organisms. If you do that in fish, you have to wait 4 or 5 years for a fish to get old. I mean, zebrafish. And mice, it's expensive to keep mice going for long periods of time. So that's how we exploit, models, and hopefully we can move quickly to make contributions in aging. So overall at MDIBL, we look for representation of investigators doing work in different models. And then we try to collate results or understand how we all interact so there's synergy between the labs. So this past year, we've recruited a new investigator, Prayag Murawala, in limb regeneration. And he's basically brought the axolotl up to speed in terms of genetic manipulations and genetic lineage tracing or tracking cells through regenerative processes. So that's a great contribution. Another new faculty joining us this spring is Romain Madeleine, who's identified neural stem cells in the zebrafish. And taking a pretty similar approach to what I've been doing is how do these stem cells get activated? How do they communicate? What are the differences between humans and fish, and how can we bridge the gap? So that's really our general focus with these broad areas of regeneration and aging. And we seek to continue to incorporate students in all this work. I have an intern this year who decided in the face of the pandemic and basically the transition to online education for everything, that wasn't all that appealing to her. And she decided to join our lab. And so she hit a pretty steep learning curve, but she's really become a productive member of our community. So we incorporate students at all levels, really, in engaging them in science. And I think it's a win-win. I mean, they gain great research experience and we gain a really motivated young investigator. So in terms of the mission of MDIBL, education remains one of our core missions. And I think it really grows the enterprise, not just at MDIBL, but when they leave and take what they've learned elsewhere. I think it really, it helps shape the whole enterprise nationally.
Well, MDIBL was one of my inspirations for starting the festival. A few people, some of your colleagues know that, because I've known about MDIBL since the early 90s. When my husband was in graduate school, a really good friend of his was in biology, and she would come up, they would do research back when it was just a summer institute up here. And we actually honeymooned at MDIBL. So part of our honeymoon was going and seeing her lab, because we are that geeky, which was funny.
Labs are fun.
They are fun, but
you know, lab.
Not a lot of people would do that, I think. So, and I think MDIBL is one of the far too hidden treasures that we have in Maine. And the fact that you're as better known outside the state than in, I have to say, drives me a little crazy. So that is one of the reasons I really wanted to talk to you. I think every time I talk to someone who's working there, I am both inspired and blown away by the science that's happening and how cool it is. And
thanks for that.
You're welcome.
I like it too.
Yeah.
I came.
I continue to marvel at all of the things that we can learn from species that don't look like us. And you know, when you explain it as well as you did, it all makes sense, right? Because when you're getting to the cellular level, there has to be things that are the same. So I really appreciate that. It makes it all make sense to me right then. So yeah. I really appreciate your time with this. More importantly, I really appreciate what you're looking at. I think it's just really cool stuff.
Thank you. It's nice to have the opportunity to talk about it all and fit all the pieces together. It's a great group, and I really enjoy the spirit there as much as anything. The community of people, we got each other's backs and we help each other out, and so it's a great institution, I think.
It's one of my favorite places to come visit and talk to people. I love the work happening there. So, Iain, I will be roping you in at some point when the festival is back in session and live and in person, you or some of your colleagues, to talk more about your science. But for now, this will have to do for us.
Well, thanks. It's been fun.
The Maine Science Podcast has received support from the Maine Technology Institute and is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is produced and edited by me, Kate Dickerson. I received production support from Maranda Bouchard. The Discover Maine theme was composed and performed by Nick Parker.